Hello to everyone. My name is Gonzalez Dahua. I am from the National University of San Marcos, or in Spanish, Universidad Nacional Mayor de San Marcos, back in Peru. And this time I'm going to talk to you a little bit of my research experience, the one that I consider the most important so far. So I work in a molecular biology lab back in my university, and my work, which is also my thesis project, is mainly focused on finding mutations on the genome of the hepatitis B virus. So just to explain a little bit of the problematic of this work, I have to mention that there are several genotypes in the hepatitis B virus. We have genotype A, genotype B, and so on until the letter H. And all of these genotypes are different among each other, and they have different abilities. So for instance, the hepatitis B virus genotype B is by far more aggressive. People tend to develop cancer faster than any other genotypes. Besides, they are differently distributed around the world. So for instance, the genotype B is mainly focused on Asia, and genotypes H and F are mainly focused on South America, especially the F, which is focused on Peru, is prevalent in Peru. However, so far there are many studies that proves this, even though that Peru is considered an endemic area of this virus. So my objectives in my thesis are to make a phylogenetic study in order to prove or confirm the prevalence of these genotypes and to support any other information that says this. My second objective is to find mutations associated to resistance in order to make an epidemiologic study. Because the vaccine has been here in this country for more than 20 years, but there hasn't been any monitoring so far. Well, working in any project actually gets you to different challenge and problems, and this hasn't been an exception. I guess that my first challenge was to take the theory to the practice, because it is, even though that it may sound easy, it is not. So for instance, you may know all the theory behind a PCR assay. You may know that, okay, you need this enzyme, and this whole process works like this. But if somebody tells you, hey, could you standardize a PCR assay, then that's another thing. Then you start thinking of, okay, what about the cycling? What about the melting temperature and many other variables that could affect your results? You have to learn on your own, you have to start reading, and many other stuff. Or what about if you have inspecificities in your PCR assay on other bands? What do you do then? So those kind of things that actually are different from the theory, but you have to learn them in order to overcome your problems. And the second challenge, I guess, is that sometimes you are so submerged in your own world, in your own research, that sometimes you forget about little details. So for instance, if your PCR doesn't go out as you may think, could be that your kit is contaminated. It happened to me. I had to repurify all my DNA, but it took me 1 month to realize that, and 1 month is a lot of time when you're doing research. And I believe that all of this experience made me to one conclusion, the conclusion that I actually learned by the hard way, I guess, that is commitment to yourself, because you are doing research. It is not a course that you're taking at university, that you only wait for the semester to end and pass the course. It is your responsibility, and it is the same responsibility that gives you the satisfaction that when you got your results, they are because of you. It is not because of somebody else. My name is Gonzalez Dahua. I'm applying for the REPU program 2019, and thanks a lot. I hope to see you all soon.